Hello, everyone. On behalf of Carterra and Biointron, I would like to welcome you to our webinar, Innovative Methods Advancing Discovery and Characterization of Antibodies. My name is Cherie Salazar, and I'm the host and moderator for this event. If you have any questions you would like to ask our speakers, please go to the bottom of your screen and click on the Q&A button, then type in your question. We will get to the questions after the presentations. I'd now like to introduce our speakers. Dr. Long Chu is the R&D Director at Biointron, and Dr. Dan Bettinger is the Senior Manager of Field Application Science at Carterra. We will start the webinar with Dr. Chu. Dr. Chu, please go ahead.
Thank you, Sherry, for your introduction. Good evening, everyone. It's my great honor to have the chance to present here. Today, I will talk about the new technologies and the platforms to empower antibody discovery. And over the past 3 decades, the development of antibody drugs is promising and rapid. We are seeing more and more antibody drugs entering the market to benefit human health. 2021 saw FDA approved hundreds antibody drug. So now we have entered the era of 100+ antibody drugs. And the trend looks very encouraging, but the antibody drug industry has been troubled by a number of bottlenecks, such as big size, poor tissue penetration, and the difficulty in modular coupling, and immunogenicity problems, no affinity, and so on. These bottlenecks call for new technologies and platforms to break through. And we believe single domain antibodies offer new options in antibody drug discovery. As we all know, in some families and shocks, there are some heavy chain only antibody with light chain missing. The variable domains of these heavy chain antibodies are often called VHH or narrowbodies. They have a lot of unique advantages, such as small size, good tissue penetration, high stability, and great potential as modules for downstream engineering, good solubility and high homology, and a flexible production system. They're so good. And then how should we use VHH? Okay, here just to show a simple workflow. And usually we start with alpaca immunization to generate the antibody library. And then we screen hits from the library. And then a lot of functional assays performed to identify lead antibodies with good potentials. Then these lead molecules are subjected to optimization, like homogenization and affinity maturations. After that, they are ready for the downstream applications, say the construction of bispecific antibodies. There are many methods for the antibody screening. And today I just talk about phage display and single B cell sorting platforms. At Biointron, we have abundant experience in VHH discovery. We have our own alpaca range with over 300 alpacas. And this picture demonstrates the phage display workflow. And the starting point is the immunization of an alpaca, and then PBMC is isolated. And then the VHH genes got amplified and inserted into PhageMES, and then transformed into competent cells for the construction of VHH libraries, and which enable a vast connection of VHH variants get displayed on phage surfaces. And the phage library usually have a pretty big capacity, say usually more than 10 to the 9th power. Okay, then how should we screen hits? Usually we perform several rounds of biopanning. And after each panning, the positive phage that can specifically bind to the antigen and get isolated and amplified. Usually after 3 rounds of panning, the positive rate in the library can achieve double-digit percentages. At this point, we can pick up monoclonal phages from the library and then perform ELISA assays to identify good positive phages and then send them for sequencing to obtain the VHH sequences. Next, I will use an example to show the process. And in this case, the antigen is called X. It's a receptor expressed on the surface of T cells. Okay, the alpaca immunization and construction of phage library is pretty straightforward. I just go directly to the panning stages. And here we use both solid phase panning and liquid phase panning. And in solid phase panning, and after 3 rounds of panning, actually we picked over 1,000 clones. And actually we find out that 360 are positive clones, which result in 11 unique sequences. And in liquid panning, the results are similar, and we finally got 900 something positive clones, and which resulted in 43 unique sequences. So totally, we obtained 54 unique sequences as our hits. Okay, so the process usually take more than 4 months, and many people may say, okay, that phage display approach is good, but it's still very time consuming. Can we have a more powerful platform and cut the time by half? Actually, at Biointron, we have the new single B cell platform called AbDrop. Yeah, it can work as an ultra-fast antibody sorting platform and accomplish and get the job done with reduced time. We reduce the timeline. Okay, and actually this just to show, okay, the conventional work process here. Actually, many steps can be replaced by this AbDrop system, which can got this tedious job get done within 1 day. So the totally the timeline can be greatly reduced by 2 to 3 months, which is significant. And this slide just to show how the platform work. And actually it can generate tiny volume oil droplet, and each droplet can contain 1 B cell together with antigens and then pass through the detector 1 by 1. The detector can tell us if the oil droplet contains a good B cell or not. If they are good, these good B cells got pulled together and then sent for sequencing to obtain the candidates sequences. This is just one example, and after one experiment on the AbDrop platform, we can obtain the 100 of sequences. And then we use Carterra to perform the affinity test to identify their affinity, and yeah. And in actually this case, we are doing the anti-PD-1 antibody discovery, and the results is pretty good. We obtained 62 hits, and the affinity test turned out that most of them show very good binding. And we all know that there are many PD-1 antibody drugs on the market, and we are curious if our hits show the similar binding epitopes with any of these anti-PD-1 antibody drugs. So we performed the epitope binning on Carterra. It is very powerful application. Actually, say if we have say 50 hits, the Carterra system can perform the 50 by 50 competitive binding experiment, and then the 50 hits can be categorized based on the similarity of their binding epitopes. Okay, so here's our results, and we performed the epitope bindings with two bisimilar, and the results is pretty interesting. We found out that 19 hits show very similar epitopes as one of the bisimilars, but none of our hits have the same epitopes as the other one. Okay, okay, I have finished the introduction of phage display and single B cell platforms, and then next step is the functional assays on hits to identify the good lead antibodies, and we begin with ELISA assays, and okay, the binding are pretty good. We also perform the HPR test on Biaco 8K. Okay, the HPR results are also pretty good. Okay, so you see, we use both ELISA and Biaco to perform the affinity test. They both have advantages and disadvantages. Say, ELISA are cheap and convenient, and it can work with supernatants. And Biaco can provide very accurate affinity data, but the instrument is very expensive and the operation is complicated, and also they have higher requirement for samples, and also Biaco is not high throughput. Thus, we wanna have a new option that can combine the advantages of both. And fortunately, we have the Carterra can make up for those shortcomings. And Carterra is high throughput, and it can work with supernatants. It can provide a good K on K off KDs, and the data analysis is very powerful and broad. So Carterra is widely used at Biointron in antibody discovery and characterization. And here just to show, and the Carterra can be used at every stage of antibody discovery and characterization. Here, the Carterra is used to perform affinity test, over hundreds of antibody hits at a time, which can save a lot of time. Okay, besides affinity, we also want to know the blocking ability of our hits. And here we performed the ligand blocking assays, and the results showed that 44 have clearly ability to block the interaction between the ligands and the antigen X, which is pretty good. Okay, and so after these functional assays, then we identified several lead molecules, but we know that most of VHH have low affinity, so affinity maturation is a must. At Biointron, we have the FCMES platform, which called the full coverage mammalian expression system for affinity maturation. And it uses a site-saturated mutagenesis in each site in CDR regions to introduce the sufficient mutation diversity. Basically, okay, for each site in CDR regions, the original amino acid is replaced by 11 other 17 other amino acids to generate a site-saturated mutant plasmid library. And then this site-saturation mutant plasmid library transformed into combinant cells, amplified and extracted, and then expect into mammalian cells for antibody expression. And usually we end up with thousands of supernatants, okay, expressed by thousands of single point mutant antibodies. With these supernatants, we perform the high throughput ELISA assays to screen for mutation hotspots. Hotspots refer to the single point mutations that can significantly improve the binding ability compared to the parentals. Okay, then we can obtain a lot of useful mutation hotspot informations. We select the good hotspots and do the combination to design dozens of candidate antibody sequences, which are expressed and characterized by Carterra and some other tests. Okay, here just as examples. We know that say some VHH their K-on need improvement, but say in the right one the K-off is too high. Okay, so we can improve their affinity and special and respectively. Say in this case, we significantly improve the K-on and result in more than 100-fold increase in affinity. And in this case, K-off is too high. With just 4 hotspots combination, we achieve, say, as high as about 23-fold increase in affinity. Okay, so after this optimization and then the VHH, the sequences of the lead molecules are ready for the final applications, and at Biointron, we are very experienced in construction of a variety of bispecific antibody formats. Okay, so I will just give a quick introduction to Biointron. We have centers in China, in the United States, and Europe, and we are focused on the antibody production, discovery, and optimizations. And we have state-of-the-art facilities and also build very advanced automated workstation, which can enable us to provide very reliable and fast deliveries. And so that's all of my presentations. Thank you. And now I turn it over to Dr. Bellinger.
Well, thank you, Dr. Xu. Let me share my screen here. Okay. Hide floating point controls. All right, well, thank you, Dr. Xu. So today I'm going to talk about some new approaches and applications we have for the LSA and Carterra's Ultra platforms, including how these approaches can apply to multi-specific antibodies. So I like to start by introducing the Carterra ecosystem, as I like to call it. We now have 3 instrument platforms: our original LSA, the sort of updated, more sensitive LSA XT, and our new Ultra platform, which has an emphasis on small molecule binding applications and just general higher sensitivity. We also have a software environment that includes 3 software packages: the Navigator software that we use to design experiments and collect data. Then there's 2 analysis software packages that, they all come together as a bundle. One is called Kinetics and one is called Epitope, and it's relatively self-explanatory what those focus on. And then we have a whole line of biosensor chips and consumables that you can use to run these assays. So the LSA platforms and the Ultra are array-based SPR systems. And these are really ideally suited for a lot of the core applications in biologics and antibody drug discovery, being kinetics and affinity analysis, competition-based epitope binning, and peptide mutant mapping and quantitation. And in addition to having a hardware architecture that makes all of these assay formats both very high throughput and not consumptive of sample, we put a huge amount of effort into designing analytical software packages that make processing all of this data fast and rapid, but also visually rich, including features like in our epitope analysis software, we have these heat map and network plots that can present a lot of information in a concise way. So in this talk, I'm going to talk about the LSA XT and looking at Fab kinetic analysis from crude periplasmic extracts. This is where we can measure the full on rate, off rate, and affinity of these without the need for purification. We'll talk a little bit about epitope binning and its role in bispecific or biparatopic antibody discovery, and then also talk about some assay formats that are more specific for bispecifics, including independence or non-interference of each binding site and bridging assays. And then I will also introduce the Carterra Ultra, which is our new platform and enables the binding screening of small molecules and fragment libraries to many targets simultaneously. So the LSA and Ultra platforms both use a similar architecture shown here, where we have two semi-independent fluidic systems. The 96 channel side is used to flow 96 samples at a time on the surface. This is used typically for array preparation or capture. And we can do in the LSA 4 prints of 96 to create a 384 array, or on the Ultra, it's 2 prints and 192. And then the 96 channel mechanism can move out of the way, and a single flow cell will dock over that entire array area and do serial injections, either of different concentrations of antigen for kinetics or of competitors for an epitope binning. This is what we call one-on-many analysis, and it's what gives these platforms their really high throughput and low sample consumption. And talk about this in a little bit more detail. This is an animation of the 96 channel side of the instrument. So you can see that the 96 needles will descend down into one of 3 microplate locations and draw the sample to the chip surface and flow it back and forth. So if this is a capture where we have an affinity-based surface, we can flow those samples back and forth over the surface for a long period of time. 40 minutes is not atypical, and get a good enrichment of the antibodies or whatever the ligands are in the samples to the chip surface. This is allowing for long contact times under high flow rate without having to increase the sample volume or decrease the flow rate again. So I'll run this video again real quick. If you do this, once for 96, then you can go wash the needles, get additional samples, and do up to 4 of these on an LSA to create a 384 spot array, which is shown in the last image here when the flow cell undocks. It's a higher density array where the injection or the spots are interlaced. Then the single flow cell can dock over that entire area where the array is printed and flow 1 sample. So again, a 270 microliter sample volume gives you binding information across the entirety of the array. This is what allows us to do this one-on-many analysis and gives the real efficiency. So, 1 injection per concentration on a kinetic titration, or 1 injection of a competitor in an epitope binning will give you sandwiching information for an entire array of clones. So we'll move right into an example here of this is the capture of fabs out of periplasmic extracts, which is a phage panning workflow. So you can induce an expression of the fabs into the periplasm and then create extracts from that. To do this, we used a streptavidin sensor chip, an HC30M chip, and captured the anti-CH1, which is a fab constant region domain antibody that we purchased from Thermo. It's actually a VHH antibody. So it is a nice monoclonal for capture. We inject that over the streptavidin surface, create a lawn of about 1,200, 1,300 RUs, at which point then we can flow the 96 well plates of fab samples. So here you can see 3 captures. These are crude samples, so relatively straight bacterial extracts. So we get a large bulk refractive index shift during the flow. And at the end, when it returns to buffer, there is a tiny amount of Fab, about 75 RUs, captured onto the array. These are very low expressing samples, and you need a long capture time to get enough enrichment to the chip surface to be able to see good kinetics. This is actually a 40-minute flow of those antibodies to capture them. But that is long enough to where most of them achieve similar levels of immobilization, and we can get good kinetics from that. So at that point, then the single flow cell will dock, and we will do a series of blank injections to stabilize the surface, and then an increasing concentration series of antigen. And that is shown here, where we're going from 1.9 nanomolar to 500 nanomolar in a four-fold series. And from this, we can fit the on rate, the off rate, the Rmax, and the affinity using a one-to-one Langmuir model. So this is double reference data at this point. And if we look at that in aggregate, this is showing the 96 clones captured to the array in triplicate. So this is a single run. This entire analysis used 7.8 micrograms of antigen to generate all of this kinetic data, because remember, it's one injection of each concentration to generate all of this data in parallel. There's some highlighting here in the data. So spots with very low signal are called non-binders and shaded gray. Things that are purple have a kinetic limitation where the antigen concentration wasn't high enough to drive towards saturation and give a well-described Rmax, so that is flagged. And then things that generally have high error in the fit are flagged in yellow. So there's some automated QC profiling of the data in this. And if we zoom in on some of those, so this are two different antibodies, each captured in triplicate. You can see we're able to get well-resolved kinetic profiles with full kinetic information from these. And on the XT, this can be done at a relatively low signal level. These are less than 10 RU sensigrams for all of these, and we're getting consistent kinetic estimates across the three replicates, even at the lowest. The third replicate, these are the third time these samples were actually captured onto the array. We started seeing a little depletion or dilution of the samples. So signal's a little smaller, but it's still giving us the same kinetic result. So we were able to get triplicates from a single well of bacterial extract in that test. Okay, moving on, we'll talk a little bit about epitope binning. So the Carterra platforms really enable pairwise competition-based epitope binning at a new scale. We do this by creating an array of antibodies. So you can put up to 384 unique antibodies on an LSA array, 190, I guess, on a Ultra. And then we use two assay formats. One we call classical sandwich, and that is where you inject antigen followed by an injection of sandwiching antibody and then regenerate the surface and repeat for each clone. So you generate pairwise data that way. If the antigen is multivalent, a premix approach is used where you mix excess competitor antibody against a fixed concentration of antigen and inject that over and look for either sandwiching or a loss of that. So after those runs are completed, we open the data in the epitope software, and we have some great visualization and tools to do this processing in there. So shown here is a classical sandwich data set. This is from one spot. So you can see there are many cycles injected over it. So one antibody in the array where we have an antigen injection. The green bar is normalization. So it sets the binding level to 1.0. So even if there's some variance across the assay, it scales everything equivalently. The second report point bar here is the report point, and that is actually the value that will populate the heat map table. So you can see the blue traces are buffer control injections. And so the value that is populated into the heat map is actually the difference from the proximal control. So even if you get a significant amount of dissociation of the antigen from the captured antibody, because we're doing it the analysis is looking at the normalized signal relative to the controls, we're still able to differentiate that, often using a global cutoff. So once these data are set with the time points and the cutoffs, The heat map is populated. In this table, the rows are immobilized ligands or antibodies, and the injected columns are the columns are the injected analytes. If it's green, that means that these interactions are sandwiching. If they're red, they're blocking. So we can create this pairwise map of the way these interact, and the software will automatically sort these to cluster like bins together. It will generate dendrograms to show how these relate, and then construct these network plots. In these network plots, every clone is a node, either a circle or a square. If a line is connecting them, that means that they are competitive with each other, and there's a blocking relationship. If they're contained within one of these colored regions, we call these epitope bins or communities, depending on sort of the level that you're analyzing the data at, and they mean that those clones share a near identical or very similar binding profile in the heat map. So looking at this a little more, again, the immobilized antibodies are the rows, the injected antibodies are the columns. This is an example of a large single epitope binning experiment. In this case, there's 170 immobilized antibodies that were able to be included in the analysis and 188 analytes. This is so about 32,000 interactions are shown. And this is a good performing assay that's pretty representative of the way you would like these to look. We have the self versus self being highlighted in the black, and they're all competitive with each other, which is good. And we have clear symmetric behavior where we're having similar patterns of competition in both orientations when you look at it. And the software can cluster these, and we can interpret these to a relatively fine resolution of epitope behavior. So here's an example of a community where all of these clones are competitive with each other. They share many blocking and many sandwiching relationships. So this is a nice, coherent community that is very descriptive of this epitope behavior of these clones. Here is a neighboring bin or community, and you can see they're also competitive with each other, but also competitive with the clones from the previous bin, and they share many sandwiching and many blocking relationships. But down here and over here, there's a difference. These are sandwiching with clones from this community, whereas these are blocking. So we've now included this to enough clones and enough diversity in this where we can do fairly fine resolution differentiation of shifted epitopes, or, so these are overlapping competitive epitopes, but they are different in that they have sort of moved around the receptor. And you can continue going through the data and categorizing these different activities. Here's another overlapping epitope bin that actually has a little bit more differentiation, has additional blocking, and then a different behavior versus subsets of these over here. And you can kind of walk through the entire data set, establishing these communities and these patterns of shared behavior that identify at a high resolution these epitope clusters. So how can this be applied? So here's a published example from Science Translational Medicine. Josh Tan's group at NIAID did this. They were trying to make therapeutics to SARS-CoV-2, and they identified a number of potent antibodies where they did affinity characterization on the LSA and kinetics and epitope binning. And they found that they had some potent neutralizers from a number of bins and they wanted to try to make cocktails that may have synergistic potency by neutralizing different epitopes. When they did that combination, they found that really the potency advantage was additive. There wasn't a clear synergy. So they said, okay, well, maybe we can make a biparatopic using the DVD IG strategy, which was one of the formats included on Dr. Shu's slide. And so they made a combinatorial matrix of a number of potent neutralizers from different epitope bins and characterized them in this DVD IG format. Here's an interesting example of a demonstration of the actual biparatopic nature of these clones. So these are classical sandwich binning assay format using two different ligands. So these are the monospecific 503 and 664, we will call them. We get antigen binding. If you inject the same clone, it does not sandwich. If you inject either the 664 monospecific or the 503 664 biparatopic, we do get sandwiching. And the same in the inverse. So if you have 664 immobilized and injected, it blocks. If you inject 503, it sandwiches. And if you inject 503,664 with either of two different linkers, you get a good sandwiching. So this is confirmation that these biparatopics, the 503,664, actually recognizes both distinct epitopes within that. And then these were tested for potency. And there were examples from the combinatorial set that showed a 100-fold increase in synergistic potency versus the combination of two clones alone. This was looked into structurally, and it was actually a really interesting behavior where two different monomers within the RBD spike were being sort of cross-linked together, forcing the entire spike molecule, spike trimer, into a totally inactive conformation. So rather than just being a sort of stoichiometric blockade of binding, it was causing the entire receptor to be sort of knocked out with a biparatopic molecule binding it. This is sort of exactly, I think, what they were hoping to see. We'll talk now about a few sort of bispecifics. So this is assuming your binder is actually binding two different targets, assay formats to look at. So the most basic characterization would be the affinity of your biparatopic constructs, probably comparing them to the parental, single specific controls. So this is basically just standard capture kinetics for us, but because the LSA does these things in high throughput, it's actually very easy to test large panels of these. So in this example, we'll say we have 12 monospecifics to each target. You can construct a library of 144 combinatorial mutants of those, and you can run those in a single afternoon or evening affinity. This can actually be expanded on the LSA up to about 1,152 unique ligands in about a 2-day run, and you can do multiple analytes to those. So this would be an example of data from that. So if we have bispecifics targeting antigen A, these are the parental controls, and then in each row are the constructs made using that sequence. You can see that generally we get good recapitulation of binding of those in the bispecific constructs. There's a few examples where maybe the binding is impaired. And then if we look at the antigen B binding in the same array, it can be the same capture kinetics experiment. We get the 12 examples of the biparatopic. In this case, all of the antigen B binding is retained in these constructs with equivalent affinity. So this would be the desired result for that as well. So there's some other tests you can do on bispecifics. You may want to know, if I'm occupying one site, do I affect the binding of the alternative antigen? In this case, you can, if it's one of them is high affinity, you can just saturate that site with antigen and then inject a titration series of the other and get full affinity. If, say, antigen B interaction is weaker and you're still trying to measure the antigen A, you would need to do several concentrations of antigen A in the presence of a saturating concentration of antigen B, sort of include in injections and reference that out. So a little messier, but still totally doable, and you can clearly identify the binding profile of A. We have an example of that. This was a high affinity B binder, so in this case, the B is unoccupied, and we get kinetics. And then in this example, the B site is occupied, and the kinetics look largely similar, very much the same. So in this case, occupancy of B does not affect the binding of A. And then the last bispecific format is one that truly demonstrates that you can have co-occupancy of the molecule. And this is probably the only one that really totally definitively shows that. So this would be, you could say, coat the array surface with, or capture, antigen A, and then use the 96-channel mode to flow your bispecific. So they will interact with the surface and attach via their antigen B specificity. And then come in with injections of antigen A, and you will only see antigen A binding to that surface if they're binding via the antigen B specificity, which is capturing the antibodies to the surface. So this can be done at scale. of antibodies automated to profile and demonstrate the dual occupancy for a large number of clones in a single experiment. Okay. So now I'm going to talk a little bit about our newest platform that we're excited to introduce, which is the LSA Ultra. So the LSA platform was our first platform and was really well suited for antibody characterization and other protein interaction analysis, protein library screening. The LSA XT works just like an LSA, it just has higher sensitivity. This really brought us down into the range of PROTACs and other sort of larger small molecules in the 500 Dalton range. And the Ultra takes us all the way in sort of therapeutic molecular space, or at least investigational, with fragment binding, down to the 100 Dalton range. The difference in these platforms are all detailed in this table. If you look over here at the Ultra, we're getting a lower signal to noise than the LSA or XT. The data collection rate is faster. We've actually have less spots on the array. There's a bit of a trade-off there to increase the data collection rate and decrease the noise. But we now have sensitivity, as I said, down to sub 100 Dalton or 100 Dalton. And the single flow cell fluidics module is almost entirely new, where we can now rapidly process injections down to 180 microliter sample volume, where the LSA is 270, which opens up the sort of standard 200 microliter deep well plate format. And the increased speed and sample processing allows you to do 384 single flow cell injections within a day. And that can include things like 50% DMSO washing of the single channel fluidics. It also has an expanded thermal range where we can now do full kinetic analysis from 10 to 40 degrees instead of 15 to 40, like the LSA and LSA XT. So the enhanced sensitivity is demonstrated here. This is a injection or a series of kinetic analysis looking at the binding of ethane sulfonamide, which is a 109 Dalton fragment to carbonic anhydrase, which was captured to a nickel surface. These are actually duplicate injections of each concentration shown at many spots within the array. So you can see we get very low noise, very reproducible binding, even of these sort of challenging small molecules. So what can we do with this? Definitely small molecules hasn't conventionally been something where people are looking for high throughput array-based thing. And we think that there's some opportunities to assess specificity and binding selectivity in a different way with this platform. So here's an example study where the Maybridge 1000 fragment library was purchased and screened against a panel of 125 biotinylated kinases provided to us by Carna Bio. This allows you to select positive binders out of this library against all 125 kinases, and then you can run follow-up experiments to look at affinity or ligand deficiency determination. So when you run this 125 kinases against 1,000 fragments, this is a 3-day experiment, you end up with 125,000 interactions of data points. This experiment was done at 300 micromolar compound concentration in a fairly complex buffer with glycerol, magnesium chloride, DMSO, and some DTT, which is required for fragment solubility and the kinase activity in the array. This was done at 15 degrees C. And in this animation that's playing over here on the right, these are the actual fragments from the Maybridge 1000 library being shown at the effective screening rate of the platform. So if you consider 125 kinases and an injection of a fragment every 3.5 minutes, you're actually getting an effective data point about every 1.7 seconds. So this is really transformative, we think, in this idea of fragment screening, where you could do entire families of proteins against a library and understand selectivity up front. Be great for SAR applications and things like that. We have a newly developed software that helps process these huge data sets. Again, this is about 125,000 interactions. We're getting 1,000 compounds against 125 receptors. So we have these report point plots, where in this case, we were using ADP as the binding control for the kinases every 10 cycles or so. And that allows you to track the activity of the kinases over time. And we're able to see the binding positive fragments and call them out against all of these targets simultaneously. So we're talking about things like families of proteins like kinases. If you have inhibitors, you can run concentration series and get kinetics of these to the entire panel of kinases simultaneously. And so in a single experiment, you can do things like populate a kinome tree, where here we have the affinity of the interaction shown as the size of the circle on the different kinases that are represented. And we've done some of these where we can compare the affinity value to the neutralization that's done from the cell-based assays that people typically pay, third-party CROs to generate this data, and they match very closely. So taking something that was quite an expensive and large endeavor to get this kind of information, being able to run it in a single experiment in a short amount of time is potentially really valuable. So with that, I can wrap up. HTSPR is an efficient way to generate high-quality kinetic data for large panels of binders, even from crude samples. The LSA enables workflows to address special needs for binding characterization of bispecific and biparatopic binders. And then the Carterra Ultra is now enabling fragment and small molecule drug discovery screening. It has high sensitivity with advanced microfluidics, optics, and enhanced thermal performance that allows us to do this. It's flexible. It can handle nearly any sample type from fragments to antibodies. And the new advanced single flow cell architecture can reduce experimental runtimes and make the processing of these samples faster. And we're working on and have next generation software tools that make processing this really high volume of small molecule screening data tractable and accessible. With that, I'd like to make some acknowledgments at Carterra. Rebecca Rich and Tony Gianetti organized and ran these fragment studies. Tim Gurman leads our commercial team. The Fab binding data that I showed is actually a collaboration we had with Pfizer and Christopher Brandstrom in San Diego, and obviously Josh Tan at the NIH for his excellent work. And with that, we can open it up to a Q&A. I think Cherie is going to.
Yes, I'm here. Thank you to both speakers. Thank you, Dr. Bettinger, for that, and Dr. Chu for a great presentation. We will now answer as many questions as we have time for. If we do not get to your question, we will follow up with you after the webinar. The first question is for Dr. Chu, and this looks like it's a two-part question. What is the sample size per experiment on the single B cell sorting platform? And about how many positive cells can be obtained?
Thank you.
Dr. Chu?
That's actually a good question to highlight how high throughput of our platform. Typically, we load 1 million B cells in each experiment, and which usually give us, say, thousands of positive B cells. Okay.
Thank you. The next question will be for Dr. Bettinger. In epitope binning, if different antibody spots have different binding profiles, are you always able to find a global cutoff value to describe blockers and sandwichers for all clones?
No, in a perfect world, that would be the case. But really what you see when you do these, especially large epitope bindings, is really a broad range of behaviors, some with very rapid dissociation, some with very stable binding, some that have a little bit more sort of sticky profiles where there's a little different behavior versus some of the ligands. So while the normalization helps a lot to make the signals similar, they're not all going to be the same. And so one thing that's actually quite powerful in the epitope binning tool is the ability to customize the cutoffs per ligand. So it can be a bit of an effort to go through the data set and do the curation, but it really pays off in the end, where you can set custom thresholds for individual ligands, and make sure that you're making the right calls in your binning map in terms of what is sandwiching and what is blocking. So I think that's one of the really powerful aspects of the epitope binning tool, is it allows you to really address a lot of the complexity that's inherent in these experiments.
Great, thank you. It looks like this next question will go to Dr. Chu. How many-fold can the affinity of an antibody typically be increased through affinity maturation?
Okay, it really depends. The answer varies. It's a case by case. Okay, so usually for parental antibody with affinity in the nanomolar range or weaker, we can see, say, tenfold or even higher increase in affinity. But say if the parental antibody has affinity, say the KD is less than 1 nanomolar, it's more challenging, but we can still guarantee at least a 5-fold increase in affinity.
Great, thank you. The next question, this one would be for Dr. Bettinger. If an array has 384 spots, how can you screen up to 1,152 samples in a run?
Oh yeah, good question. I didn't really talk about that. So if you have a capture surface, so an anti-FC, an anti-His, protein AG, anti-V5 tag, or whatever your system is that you're using to tether via affinity those ligands to the surface is regeneratable. So you can have 384 well plates in the instrument, with high diversity of clones or samples or even replicates within them. And you can capture that 384 at a time, strip it with regeneration. If you're using nickel NTA or something, you have to recharge the surface, and then you can load additional samples on. So that can all happen within a single run. We can have many sets, we call them, of analytes or kinetics in those. If you are doing something like epitope binning, where you're covalently linking the antibodies to the surface, then you are sort of limited to the 384 spots because they don't go anywhere. They stay attached.
Great. We have time for one other question during the live webinar, and this one will be for Dr. Bettinger. How do you measure the affinity of a biparatopic binder?
That's a good question, right? Because with a bispecific, you have all these options. You can do the targets individually or various orientations or saturation link, but a biparatopic, you're both arms of that binding domain are binding to different parts on the same molecule. And if it's working right, they do so simultaneously. So those affinities are essentially always avidities. Like the combination of the combined affinity. So in that case, it's almost more of a profiling than a tight or discrete kinetic description of a one-to-one binding, because it is not a one-to-one binding. So it's sort of the conventional understanding of Ka and Kd don't truly apply. So I think if you have knowledge of the kinetics of the individual binding components and you inject the biparatopic over the analyte or the analyte over the biparatopic, you should see a significant difference in that binding affinity, probably a much more stable binding, but it may not be a totally discrete affinity. Although very often a biparatopic will look like a one-to-one interaction, it is just much higher affinity than the individual components. So oftentimes they may behave that way because there isn't the interaction can saturate, not like if you were doing an avid analyte over a surface. It's a little complicated, but yeah, it is an interesting phenomenon where oftentimes comparing them to their parental controls is important to demonstrate that there is the enhancement of that binding affinity and increased stability. But it may not fit an exactly one-to-one interaction because it is a sort of different entity at that point. But unless you have sort of truncated antigens where the binding domains can be accessed discretely, like chimeric antigens or clipped or truncated forms that only access one or the other binding sites, you might end up with complex kinetics.
Okay, thank you. I would like to thank Dr. Bettinger and Dr. Shu for your answers. They were great, but that's all the time we have for questions. As I mentioned earlier, if we didn't get to your question, we will follow up with you after the webinar. Thanks to all of you who attended this webinar. If you have additional questions, you can follow up with us at questions@carterra-bio.com. A recording of this webinar will be available on demand on our website tomorrow. Thank you again and have a great rest of your day. Bye-bye now.